Hello and welcome to Adaptive Immunity. This is the third podcast in the series on adaptive immunity. This podcast is dealing with antigens. So if you look in the bottom right side of the screen, you see a bacterial cell, and then there's a sort of a shaded spot. That's the antigen, and then living in the antigen are proteins. Usually they're proteins called antigenic determinants. So the antigenic determinants are on the antigen. Antigenic determinants are also called epitopes. So it's the same thing. If you see the word antigenic determinant or you see the word epitope, it's the same thing. It's that shape that lives on the antigen. And then also in this cartoon you can see that antibodies recognize antigenic determinants on the antigen. It could also be said that antibodies recognize epitopes. And if you remember from the overview podcast, we said that B cells are the cells of the adaptive immune system that make antibodies, and then the antibodies recognize the epitopes. So your adaptive immune response, so those B cells and T cells, don't recognize a whole pathogen. So the B cells in this cartoon wouldn't recognize the entire bacteria. The immune cells would just recognize the antigenic determinants, aka epitopes. And also remember, our own cells have antigens in their cell membranes, but they're self-antigens or autoantigens. These antigens on a bacterial cell would be a non-self antigen. Antigens are not only included in cell membranes of pathogens, but also toxins can be viewed by the immune system as antigens. So some properties of antigens. Your body, the cells of your immune system will recognize the antigens by the 3D or three-dimensional shapes of their epitopes, aka antigenic determinants. A larger molecule, like 5,000 to 100,000 Daltons, that's just those are the units of measurement for a protein, Daltons. So larger proteins are better antigens than smaller proteins. But not only proteins can be antigens. You can be a glycoprotein. That could be a good antigen. So a glycoprotein is just a big protein with carbohydrates or sugars sticking in it. Lipids, so that's fats, those can be antigens, but they're less antigenic than a protein antigen. If you're a really small molecule, then you would make a poor antigen. But if you are a small molecule, like penicillin is a small molecule, you could be called a hapten. And if you're penicillin, you're a hapten. If you find a large carrier to bind you, then you, as a hapten, could become antigenic. So this is a cartoon of a protein antigen, and it shows you, so the pink line would just be the amino acid sequence of a protein, and then at a certain amino acid sequence, the body would recognize that sequence as an actual epitope. So your B cell or T cell would recognize that piece on the protein antigen called an epitope. So there's epitope 1, 2, 3, and 4 on this protein. And then if you look at the right-hand side of the screen, you can see that antibody 1, so that's the Y-shaped molecule, antibody 1 is binding epitope 1. Antibody 2, that's the Y-shaped molecule, is binding epitope 2. And antibody 3 and 4 are binding epitopes 3 and 4 respectively. If you are a larger molecule, you just have more epitopes on you, so that makes you more easily recognized by the cells of the adaptive immune system, which are the B and T cells. Some properties of antigens. There are components in a bacterial cell wall. Remember, the cell wall is just that big peptidoglycan layer. Remember also that microbes have capsules, pili, and flagella. So remember, the flagella are just those components that allow the bacteria to swim or to move. External proteins in a virus or fungi or protozoa. So remember, some viruses have coats and some don't. But any of those things in the cell wall or cell membrane that can look like an antigen to an immune system, you would call that an antigen. So if you look at the cartoon, they show a cell with a nucleus, and then they cut out a piece of the plasma membrane and enlarge that. And you can see the lipid bilayer. And then in the lipid bilayer, aka the cytoplasmic membrane, you can see the epitopes or the antigenic determinants on the antigen. There's different types of antigens. There's exogenous antigens. Those would include the toxins that you might encounter in the environment and other secretions that microbial cells might release. Endogenous antigens, those are bacteria or viruses that would reproduce inside your body's cells, and then they would make endogenous. So endo means within, endogenous antigens means that the antigens are being made inside the cell. So if you look at the cartoon under letter B, it says exogenous antigens. So it shows two different extracellular microbes, could be a bacteria or a virus, and then the exogenous antigens that are attached to that microbe. And then on the right hand side of the cartoon, there are endogenous antigens. So it shows that you have an intracellular virus in an infected cell, and then the body will chop up that virus and display the endogenous antigens on the cell surface. Remember, if you listened to previous podcasts, we talked about autoantigens. Those are also called self-antigens. So every eukaryotic cell in your body has antigens on it, and the immune system recognizes those antigens as self-antigens. Those are normal antigens, and your B cells and T cells should not attack those eukaryotic cells that have self-antigens on them unless you have an autoimmune disease.